Hello, I'm Dongyoung Kim from South Korea. I am currently an undergraduate student intrigued by the most wonderful organism in the planet. Here I'm going to be talking about my study, how I tried to overcome the species limitation problem observed in Korean endemic harvestmen. Now I'm going to start. These are what I'm studying, family Paranonychidae. They are Opiliones, also known as harvestmen. They belong to suborder Laniaturets, having developed exoskeleton and relatively short legs. Family Paranonychidae is a very interesting taxon in the aspect of biogeography because it shows amphibaryngian distribution. And it is known that they show conservative morphology derived from strong ecological constraints. They are microhabitat specialists, and it is noteworthy that they have low mobility and high regional endemism, strong population structure. Among the genera, here, genus Kaolinonychus is an endemic one to Korea. It is currently a monotypic genus, so there is one species, K. koreanus, containing two subspecies. In order to meet them, I have been exploring caves and mountains with cool and moist conditions like this. And in there, I could find them flipping up some rocks or logs. Their appearance is like this. Their size is about 1 millimeter, so they are very tiny harvestmen. Throughout my sampling and specimens, I got to know that they are found in almost every place on the mainland of South Korea. And taking into account their already known ecological features like low visibility, I've come to conceive a question that all of these populations are really one same species. So I wanted to review their classification, and this is why I started this study. But the problem was not pretty easy. There are some decisive problems, and first of all, in morphology. In traditional taxonomy of lots of invertebrates as well as harvestmen, the genital morphology is very important for species delimitation because it can show reproductive isolation intuitively. It is rational and extremely useful in lots of cases, but in Kaolinonychus, they show strong conservatism in male genitalia. So based on reproductive organ, they were overlumped into one species. On the other hand, DNA barcode analysis highly oversplitted them. According to the universal delimitation algorithm like ABGD, there are 16 species in Kaolinonychus, and it is pretty unacceptable under our biological intuition. To sum up, problems at both extremes are observed simultaneously in Kaolinonychus. So, how could we approach this problem? Without that, integrative taxonomy is required rather than relying on either side. And for integrative taxonomy, I collected morphometric data. So I measured their somatic length measurements as many as I can, and I could get 65 variables metrics. And then I also gathered CO1 sequences of mitochondrial DNA with 200 SNP loci. And using both data, we progressed discovery and validation process. Let me elaborate on the integrative taxonomy. It is based on a more practical and operational species concept and tries to examine some cohesiveness among monophyly, genetic cluster, fixed phenotype, and biological inferences we have instead of some specific analysis. And by doing so, it pursues a more quantitative and reproducible classification. And in order to do this, we have to take two stages. The first one is discovery process. Here, we classify high-dimensional data without our priori and inferences. And then in validation process, we test this hypothesis using our biological information. And we could think that what is required in each process fits very well with the task that supervised and unsupervised machine learning do, respectively. And there are published studies by Durkabedian et al. Demonstrating these ideas, so we progressed the study following these papers. First, in discovery phase, we need to establish the hypothesis from the data itself. In this process, we should use all the variables, all the characteristics, instead of selecting some specific trait under our judgment. In this way, we can build high-dimensional spaces composed of variables and characteristics, And we can figure out the unique on distribution or specific location of each population on our hypervolume. This is what we want. But the problem is that our eyes can only see up to 3 dimensions. So we have to rely on dimension-only reduction. In addition to find the location of populations, we want to see how populations are grouped in objective way. So we're going to use clustering methods. And doing these two things that we need is the main work of unsupervised learning. So this is why we actively used it. Let me tell you the result of my data. First of all, we reconstructed the phylogenetic tree and set some identical subclades as the putative groups being subject to analysis, like this. You can see that each represents a subtle difference in morphology. And then we applied various unsupervised learning analysis. First, it is the result of using CO1 data. From widely used PCA, Tistony, Random Forest, DAPC, and VAE, a variational autoencoder, a deep learning algorithm which is utilized in microbiology field in recent studies. Except VAE, we did the same tasks using morphometric data, like this. And from these results, we were able to establish the species hypothesis, suggesting that there are 3 species. After this discovery process, we can test whether our model, our hypothesis, actually fits biologically through the validation process. And in this process, we can apply a supervised learning. As suggested in this paper, we tried to use the own natural history of organisms in supervised learning-based validation. The program for validation we used is called CLASE. This program basically assumes a 2-species model and calculates the pairwise population genetic statistics for every population pairs. And then we conducted the support vector machine using these statistics data. Let me walk you through this more intuitively. A support vector machine is a supervised learning algorithm that classifies the category of unlabeled data that we don't know the answer using a classifier called support vector, which is already trained by labeled data. In our case, first of all, we can imagine the space in which each axis consists of the parameter of pairwise statistics. And here, each point becomes a population pair, not a single individual or a single population. And now we can do the training using labeled data that we already know the answer. We can find a suitable classifier, hyperplane, which can optimally divide the category of population pairs of the same species and the different species, like this. And then when we come to get a new unknown data, we firstly can map new data in our hypervolume space. And after that, we can predict which category the pair belongs to, same species or different species, using our already trained classifier. This is the way how it works. And in this CLASE paper, they suggested that they made a fine classifier that's generally available, conducting simulation analysis under various evolutionary genetic parameters. And they said it worked well when it was applied to birds, primates, and mosquitoes. If so, I wondered if it will be also suitable for our Kaolinonychus data. Unfortunately, no. Like other universal delimitation algorithm, it oversplitted each clade into different species. This is probably because the evolutionary genetic statistics observed in Kaolinonychus might not be contained in the scope of their simulation data. So, what we thought was that different ecological features inevitably have different evolutionary histories and different underlying genetic patterns. It is obvious that the microhabitat specialists with strong population structure have different natural history and genetic patterns with birds, primates, and mosquitoes. So, what we wanted to do was making a new classifier reflecting their specific ecology, natural history, and underlying genetic patterns. The reason how this idea was feasible was that within Paranonychidae, there is a genus with a well-established classification showing congruence between both genetics and morphology. It's a genus Paranonychus, studied by Dongyoung and al. So we were able to create a new classifier using genetic data of Paranonychus, because we already know the answer, which population pairs are the same species and which are not. But in here, I want to mention that there was a technical problem when I used all the data in Paranonychus. I could not figure out the region and I could not solve it yet. So as measures, I made a training set using some clade of Paranonychus. But I want to emphasize once again that this was trial and preliminary result and not the complete result. I'll try to fix this problem and progress in advance in near future. Anyway, these some clades of tree also have a similar ecological feature and genetic features to Kaolinonychus, so it could be applied with the same logic as I explained. So I could make a classifier using them. So instead of a general classifier, we predicted the species using a classifier that reflects our biological information, ecology, and genetic patterns. From this, we could obtain a delimitation result for each population pair. And putting it together, we can get the result consistent with our hypothesis. This is a much more acceptable result than the result saying that they are all one species or there are 16 species. So in my preliminary study result, it seems that there would be 3 species within the genus Kaolinonychus. 2 of them are elevated from existing subspecies and 1 may be a candidate of new species. As I said, in this presentation, I tried to show the process, how I tried to overcome the difficulties in species delimitation issues. So in order to advance my study, there are some still problems to be solved. First, my first drawback is that I only use CO1 data. It is well known that this single locus gene must have the probability of error by genetic drift called incomplete lineage sorting, especially in a shallow depth divergence. Although this C1 gene analysis itself has meaningfulness in the aspect of DNA barcode and utility of classification, but I think this study should move toward genomic level study. Second, as I mentioned, there was a technical issue to make Paranonychus training set. In machine learning, it is also well known that small training set can cause the overfitting problem, like this. So I have to fix the issue and try to use all of Paranonychus data. And furthermore, I think that what size of the training set would be suitable for accurate prediction in our supervised learning-based validation is a problem, very important problem that needs to be further considered. Last, what I want to do is figuring out more objective way for combining the result of unsupervised learning analysis. To me, addressing these issues seems the way forward. Finally, I'd like to talk carefully about what I felt while conducting this study. First, the use of artificial intelligence in the field of taxonomy has a very high utilization value and is promising. I think the way machine learning works is very similar to the mechanism of thinking that taxonomists had when classifying organisms. So therefore, the use of AI in the field of taxonomy is expected more in the future. But paradoxically, I also felt that the field of taxonomy could never be replaced by AI. Some people say AI will replace all the work done by taxonomists in near future, but I felt it would never be happened. Of course, each analysis can rely on machine learning because it can analyze bigger data in a more accurate and faster way than humans do. It is obvious. However, making an entire framework of analysis, interpretation of result, and troubleshooting for each analysis can be done by only humans, especially those with specialized knowledge in the field of biology. In particular, as mentioned, our selection and application and making an appropriate training set should be based on biological inference, natural history, and ecological understanding of organisms. So I think this is the reason why we need taxonomists even in the AI era. Thanks for attention.